My lab is interested in brain circuits and behavior. Emotion or cognition has been linked to the limbic system, which consists of the hippocampus and amygdala. Speaking of the research topics in my lab, we focus on the following. First, neuronal diversity and function of memory in the hippocampal circuitry. Second, the amygdala and neural mechanism underlying emotional behaviors. Finally, we are interested in clinically relevant problems such as comorbid affective disorders of chronic pain. The first station of the hippocampus is the dentate gyrus, which receives information from the cortex. Information is then processed in the dentate gyrus before being relayed to the downstream brain circuits. To understand the brain circuits, we mainly investigate GABAergic inhibitory neurons. In addition to local circuit, we are interested in extrinsic projections to the dentate gyrus. For example, a subset of neurons in the hypothalamus project to the dentate gyrus. Currently, we have PhD students working on this kind of projection and to understand how long-range projections innervate the dentate gyrus and control the activity of different cell types. The hippocampus is an elongated structure in humans and rodents. We are also interested in the long-distance connections along the longitudinal axis of the hippocampus. For instance, the hippocampus has dorsal and ventral poles. Both are connected by long-distance multicell axons. The function of these long axons remains unclear. Recently, we found that the projections to the dorsal and the ventral poles are very different, and such projections are relevant to anxiety-like behavior. In addition to glutamatergic neuron, a subset of inhibitory neurons make bilateral dentate gyrus projections. We employ electrophysiology to investigate the function of these long-range inhibitory connections in ex vivo slices or in vivo intact brains of anesthetized or even in freely moving animals. Speaking of inhibition, there is a special type of inhibitory cells called VIP interneurons, which mainly target inhibitory interneurons. This is called inhibiting inhibition. We are studying morpho-electrophysiological properties and functions of this group of neurons. More recently, we have become increasingly interested in the central nucleus of the amygdala. The central nucleus of the amygdala consists of many GABAergic cells. There are several different types. A PhD student performed page clump recordings from 2 to 3 neurons simultaneously and tried to understand their cell types and function. Furthermore, we try to understand how these neurons with different gene expression make functional connections. Finally, because of my MD background, I'm also interested in brain circuits in disease. Acute pain is different from chronic pain. Development of chronic pain is associated with rewiring of certain brain circuits. Almost 5 years ago, we started to team up with the clinical team in Taipei Veteran General Hospital to study neuromechanisms underlying comorbid affective disorders of chronic pain. These comorbid symptoms include anxiety, depression, and social deficits. Using a fibromyalgia mouse model, we investigated the changes in brain circuits and discovered that two populations of neurons in the central nucleus of the amygdala undergo significant changes in their synaptic input and cell excitability. This kind of change is called central sensitization. Interestingly, our preliminary research in chronic migraine also revealed central sensitization in the central nucleus of the amygdala. However, the cell type of sensitized neurons is totally different from that in fibromyalgia.